So today, it's my pleasure to introduce Dr. Elana Fertig as our guest speaker for today's seminar. Elana is a professor of oncology and associate cancer center director in quantitative sciences at Johns Hopkins University. She's the co-director of the Convergence Institute and the co-director of the Single Cell Training and Analysis Center at Johns Hopkins. Elana is a mathematician by training. She completed undergraduate training in physics and math and her graduate education in applied math. Funny enough, she worked on mathematical models of numerical weather forecasting until her postdoc, where she transitioned into computational cancer biology. Now in her position at Hopkins, her group develops computational methods to extract and predict disease-relevant signals from high-throughput cancer genomics datasets. Recently, her group has developed the Coordinated Gene Activity in Patterned Sets method, or COGAPS, which uses Bayesian non-negative matrix factorization to identify latent factors or metapathways from genomics datasets. Elana has secondary appointments in biomedical engineering and applied math and stats, and affiliations in the Institute of Computational Medicine, the Center for Computational Genomics, Machine Learning, the Mathematical Institute for Data Science, and the Center for Computational Biology, and is a Daniel Nathans Scientific Innovator. She serves in the editorial boards of PLUS Computational Biology, Cell Systems, Immunoinformatics, eLife, and Cancer Research Communications. She's received numerous honors and awards over the years, most recently being elected to the College of Fellows, American Institute for Medical and Biomedical Engineering. It's a true honor to have Elana here today with us. And today she's going to talk about forecasting pancreatic carcinogenesis through spatial multiomics. I hand it over to her.
Thank you all so much for being here, and thanks for the invite to present to the group today. So I'm going to be talking about some recent work that we've been doing, really bringing together spatial molecular profiling along with mathematical modeling and recent techniques. Please feel free to ask questions. I'd love to be a little bit more interactive as the talk goes on, but with that, I'll dive in. Just briefly, my disclosures. So I'm in a cancer center in spite of being a mathematician and bioinformatician. So a lot of what I think about is that we're sort of in this era of a technologic revolution where we have increased spatial molecular profiling at dimensions that were never before imagined, where we can get into spatial tissue, the cellular and molecular state using spatial transcriptomics technologies, link these to multi-scale information. And then the hope in cancer research is that we can leverage these for treatment decisions by getting better estimates of what cell states and cell types comprise a tumor. So hand in hand with this interpretation, the data generation is not enough, right? We really need computational methods, and not only black box tools or sort of pipelines and workflows like Surat, but really fundamentally the invention of new computational methods to answer cancer biology, where we're trying as computationalists to answer fundamental questions about tumor evolution, cellular architecture, and regulatory biology. Many of these concepts, how we address them and how we analyze data to learn them, are fundamentally unsolved questions. And so really do need to be side by side with methods development along with data analysis as a fundamental component of modern cancer research. So as was alluded to, my background is in weather forecasting, not in cancer biology or bioinformatics. And there, a lot of what I worked on is how do we integrate high throughput data sets coming from satellites and weather balloons with mechanistic, mathematical, or physics-based models to predict where the systems are going forward in time. And I hope to present to you today how I like to think about where we can take this sort of understanding and thinking about forecasting by bringing together high throughput data with mathematical models to start thinking about, can we forecast tumors and use that information in therapeutic decision-making to try to develop better interceptions to prevent disease and develop and overcome resistance. So I'd say that piece is probably aspirational and what I hope to do for the rest of my career, but I will show the building blocks of how we're sort of hoping to get there and what we've started to do. So we're focused in pancreatic precancer. This beautiful review by Laura Wood really goes through the complexity of pancreatic precancer. What happens is in the very early stages, you start to get chromosomal alterations, somatic mutations, and epigenetic alterations in precancerous lesions. Recent work in organ donor samples by the group at University of Michigan has found that even in the healthy aging pancreas, an individual can be carrying a burden of hundreds of premalignant lesions that will probably never form into pancreatic cancer. But in spite of this, you have these footprints of these early changes there. The cellular alterations in the tumor cells are consistent with a change in the tumor microenvironment, where in precancer, there are more immune cells there. And as advanced pancreatic cancer develops, you have more and more immunosuppressive cells that block the function of the immune system. And there's several different etiologies and risk factors that arise with pancreatic cancer, either due to genetic background, environmental factors like smoking, diabetes, chronic pancreatitis. All of these contribute to the development of the disease. So the fundamental question in the field on the basic biology side and therapeutics is, how are these cellular and molecular landscapes changing over time? And how does that impact when we should intercept and biomarkers perhaps for better earlier detection? Because one of the biggest limitations in this disease type is that it's detected, unfortunately, too late for a ton of people. So to start doing this, what I think a lot of people want to do is start studying the landscape of these tumors and the precancer. What's exciting about working with this from a spatial data perspective is that when you have an advanced pancreatic tumor that's resected, several of the precursor lesions are still within that sample. Because you have such a large burden of precancer lesions, you can identify them within a concurrent pancreatic sample from surgery of the tumor. So what this gives us the ability to do is start using spatial information as a surrogate for time to say, can we compare the cellular states as the disease develops from normal to chronic pancreatitis through precursor lesions and finally advanced pancreatic cancer? So we were fortunate to work with a pathologist, Liz Thompson, to select a cohort of samples that were designed in this way. And in a study led by Melissa Lyman, along with Elizabeth Jaffe, Wanjin Ho, and Neha Zaidi, to start looking at the spatial proteomics landscape of the immune cells. And so in these spatial proteomics landscapes, what you do is you pull out different immune markers, you get them from an antibody stain, you have a panel, and then you go in and you have sort of a sense of, you do some clustering, identify the cell types that are there, or apply a gating strategy to identify the cell types that are there, and can start mapping them across stages. So for example, here in this analysis, we can see if we look at T regulatory cells, there's a relatively low density of T regulatory cells in normal samples that increases with PANM and then becomes very strong in the advanced PDAC, consistent with what's known a priori about the development of immunosuppression in pancreatic cancer. So this is a very powerful demonstration that we can use space for time. The limitation with the spatial proteomics technologies is you kind of have to go in knowing what you're looking for, right? So you have to sort of say, okay, these are the cell types that we're going to look at. So on the other hand, single cell technologies, and I'll get into spatial transcriptomics in a little bit, but when we started this project, right, what was really available was dissociation-based single cell assays. So the advantage of the dissociation-based single cell assays is that they let you measure the entire tumor, right? And you can characterize from the transcriptional profiles what cell types are there. So what do people typically do when they do this analysis? And this was work led by Ben Kinney-Koster and Jackie Zimmerman. The paper finally came out this week. So, what do? We collated a bunch of data from the public domain to start looking at an atlas of pancreatic tumors. And then you apply your single cell data, you apply clustering, and you get the cell types that are there. But arguably, if you're going to do a clustering-based analysis, there's no reason you should have bothered with the high dimensional nature of the spatial of single cell data, right? You could have gotten that from a flow cytometry or a multi-parameter flow back to working with the same proteomics data that you're working with. So what is the advantage of single cell technologies in getting this higher dimensional information? So there's this beautiful review by Wagner et al. That sort of starts giving the first sense of this, is that with single cell data, because you have such high dimensional information, right, where you have all 20,000 genes in the transcriptome measured across cells, you have the ability to go beyond what cell types are there. And you're going to have information about what are the phenotypes of the cell, where is it evolutionarily, where is it in the cell cycle, where is it spatially. All of these are going to leave footprints in the data. And then our goal as computational biologists is to see, can we deconvolve the data in order to dissect these different phenotypes and these different signatures. So in order to do this, we start applying matrix factorization. And what we realized is that if you have this high dimensional data, right, and you have multiple components, the idea of dissecting these components from the data is perfectly mirrored in the mathematics of matrix factorization. So I'm going to go away for a second from like the complex pancreatic microenvironment and, 20,000 genes, just so that we're all on the same page. In my cartoon world that I've drawn with only 4 genes on my y-axis and a few groups of cells, for example, we can imagine the ones on the left are the precancer and the ones on the right are the advanced cancer. What you can very clearly see by eye is that there's 2 dominant patterns in the data, right? One that goes up and down in your precancer, one that goes up and down in your advanced cancer. And then each gene is assigned a weight as to whether it's using one or the other of those or both. And so the idea is that we attribute these patterns to the different biological processes in the data and then can leverage the gene weights that are learned in order to annotate them back to different features. So this is going to be sort of the foundation of a lot of the analysis that we do. We have a particular tool that was developed when I was a postdoc, and we've sort of kept using it through the years, called COGAPS for this matrix factorization. And the advantage of this is that it's a Bayesian sparse model. But I'm not going to go much into that today. I'm going to focus more on the applications and how we think about using it in the context of these data sets. So that's sort of the model for anybody who cares. And I will skip that slide. So what we started thinking about is, okay, now I want to start studying the cell state transitions in my data set. And if I look at this reference atlas of single cell data, what's interesting is that the cohort that we collected has true normal pancreatic cells, a set of tumor adjacent normal cells, and then clear cancer cells and epithelial cells. And we wanted to see, can we decompose this data and the different processes occurring in epithelial cells to start monitoring the transitions between normal and tumor that we observe in the data. So when we apply this, we observe several patterns, but I'm going to focus on 2 because a lot of the others were technical. But the 2 that came out that were most interesting was within the subset of tumor cells on the left, we see very clearly an upregulation of estrogen receptor signaling and KRAS signaling that's associated with cellular proliferation in a subset of the cells. On the other hand, if we look at another pattern, pattern 7, this is one that's really associated with immune rejection, epithelial to mesenchymal transition, and inflammatory response. And that one seems to be spanning both the tumor cells and a subset of the tumor adjacent normal cells. So this led us to hypothesize that there's probably a trade-off, and we certainly see in mutually exclusive cells, between inflammatory response and EMT relative to cell cycle proliferation that occurs later. And our question that we wanted to know was, because we're seeing these transitions, And because we're just taking a whole tumor and dissociating it in this atlas, are there actually precursor lesions, and are we seeing a map of cell state transitions that we can't identify or attribute to it being a precancerous sample relative to cancer because we don't have the pathology that we're mapping it back to? And can we start using that to understand the regulatory programs? So we're fortunate that we're now in an era where there's more and more spatial transcriptomics data sets. So we wanted to ask the question again, can we map these patterns to how things change from the normal pancreatic duct, low-grade and high-grade pannin relative to what we're seeing in advanced cancer? So can we start to think about how these features go backwards in time during carcinogenesis? So Laura Wood, our pathology collaborator, very generously went back to the pathology bank and grabbed a whole cohort of low-grade and high-grade pannins of 5 of each, matched with patient-matched low-grade and high-grade. I didn't realize that it's like a 1% event and what we were asking her to do and diving back in the archives to get these samples. But we were very lucky to sort of have that study. And then Luciani Kagahara, in collaboration with 10x Genomics, led the development of a beta test of the new FFP, at the time, new FFPE-based spatial transcriptomics on this cohort of low-grade and high-grade samples to try to map this. So when we run this thing, the first thing that we noticed when we ran the analysis, and we kind of, look at the H&E, get the different regions, and then we start looking at clustering in this middle row, is that we observe when we apply the same type of clustering that you would apply in single cell analysis, it doesn't really align to any of the morphologic features in the tumor. Now, why is this? Because spatial transcriptomics is not true single cell resolution, and so you have a mixture of cell types. So Ashley Keeman and Alex Bell in my group, so Ashley Keeman is starting her lab, and she had developed this very elegant machine learning classifier where you can use the morphology of cells from the H&E imaging in order to map the gross cell types that are there in the tissue. And what Alex realized is in the FFPE-based spatial transcriptomics, because we have the H&E aligned to the spatial transcriptomics data set, what we can do is use this classifier to figure out what are the broad cell types that are there, and then count up within the spot what proportion of the spot is associated with each cell type. And so you actually have a ground truth for broad cell types as to the number that are there to enhance your deconvolution, or in our case, we decided to use it as a purification method to isolate which cells are either one cell type or another. So when we apply this to our cohort, we can start to map out the transcriptional heterogeneity that compares between low-grade and high-grade tumors. And even in this case, for example, where we have a mixed duct, where part of the duct is normal, part of it is low-grade, and part of it is high-grade, all within one duct, we can map the transcriptional changes that occur across different stages, even within a single region, and look at how the transcription changes. So now how do I get back to my patterns, right? So how do I get back? This works really well for single genes, right? But I have a whole gene expression program that I've defined through matrix factorization. So how do I start looking at this and how it relates to the spatial data set that we're seeing? So Genevieve Stein-O'Brien and Gaurav Sharma, in collaboration with Loyal Goff, we've realized together in the context of single cell data, what we can do is imagine the gene expression signatures that we're seeing as the lenses through which we're viewing our data. That's all that exists. And then if we have a new data set, for example, the spatial transcriptomics, what we can do is regress our gene expression signatures relative to the gene weights that we've learned in a factorization and a reference data set. So the approach that we developed for this is in software called ProjectR, and we think of it as a transfer learning based approach. And so what this then lets us do is take the lenses that we've learned in the data and then project and map where they're going in the new data set. So what we observe when we apply these two patterns into our spatial data is mutual exclusivity between proliferative signaling and inflammatory signaling, right? So we see this trade-off between the two. And so then the question becomes, okay, they're clearly occurring in different cells spatially, which is consistent with what we saw in the single cell data, but how does this map across stages of pattern development? And what we observe is that the proliferation pattern is clearly increasing across stages of the carcinogenesis. So we're seeing this increase in proliferative signaling as the precancer lesion is developing. But interestingly, if we look at this inflammatory pattern, it might arguably be decreasing, but more likely staying a little bit flat. So it's not really associated with the cancer progression as much as the proliferative signaling is. So the question is then what's going on, right? Like what is causing the relative difference between these two patterns? So our hypothesis was that it was probably something in the microenvironment that was different, that was regulating these two processes within these epithelial cells. So, sorry, don't know why that slide was there. So what we did is then went back to 10x Genomics and asked, okay, we want to get better resolution of the cell types that are there. And so we built a collaboration with them looking at the new imaging spatial transcriptomics technology of Xenium. And the nice thing about the gene weights that we've developed from our matrix factorization is then we can use them to define a custom panel for the epithelial cells to mark the different biological processes. So we worked with 10x to develop a custom gene panel, as well as these different, as well as a standard immune panel and fibroblast panel. And so using that, we can start to map out the immune cells. We can start to see the growth of the Pannin cells coming out from the duct. And we can map the difference between proliferative signaling and this inflammatory signaling. And what's interesting is when we look at that, the inflammatory signaling seems to be going more outwards, whereas the proliferative signaling is going more inwards. And we see that this region is surrounded by fibroblasts, right? So you have, what we're hypothesizing is that you have fibroblasts, the cells is on the edge are seeing that and able to have inflammatory signaling throughout, whereas the ones closer to the duct are further away from the fibroblasts and so able to start proliferating. And so the question then was, okay, is this just an artifact of this one pretty slide that I'm showing you, or how much does this apply across a cohort of tumors? And so in order to do that, we started going back to our single cell atlas and correlating the extent to which we see the pattern with the density of fibroblasts. So we see this inflammatory pattern is occurring only in one of the two subsets of data. It happens to be the one with more fibroblasts. And when it occurs, it is correlated with fibroblast density in each patient. So the computational biologist in me, when I see something that happens in one data set and not another, immediately thinks there's a technical artifact as opposed to biology. So the question is, how do I actually see if this is something real? Or is this just some weird hat trick or technical subset within one group of cells in the data set? So in order to isolate these effects, what we wanted to do is start seeing how do they relate in experimental models where, for example, in organoids, you can get controlled experiments where you have your epithelial cells and fibroblasts in co-culture, so you know that's all that's there. Right. And so Sam Gwynn and Jackie Zimmerman led this experiment. And we wanted to see, can we apply the same transfer learning approach to co-culture relative to monoculture to validate experimentally these patterns and whether it's a real biological process induced by fibroblasts or something else that we were missing. So we ran this co-culture. We ran single cell in one of these and then a panel with bulk RNA-seq. And of course, I wouldn't be talking about it if it didn't work. But in both cases, what we saw is an enrichment in the co-culture for this EMT-like and inflammatory signaling in the co-culture relative to the monoculture, leading us to hypothesize that we really do have this fibroblast association inducing both EMT and inflammatory signaling. And Jackie and Sam went back and did a tremendous amount of experimental validation to sort of look at the implications of this, which I won't talk about. But I hope this sort of gives the sense of the power of discovery and atlases, but really linking them back to experimental data so that we can get the whole bench to bedside and through mechanistic validation. So what's the next? Yes?
There's a lot of data in the last few years that say that fibroblasts in pancreatic cancer are not uniform, that there's different kinds. I wonder if the analysis that you're doing allows you to separate, and the studies that are done here are obviously in co-culture, but in vivo, can you distinguish those different types of fibroblasts in terms of what you were just talking about?
Yeah, we can look at the different, you can certainly cluster and look at the cell-cell communication between different subsets of fibroblasts. That's something that we have a student working on now to sort of get at more refined. Unfortunately, in the organoid culture system, we're seeing that it selects for one of the subtypes. And so we don't really have the ability to mine that as well as we would like. But yeah, we definitely can. And then I sort of buried it under the rug. I didn't talk about it here, just in interest of time, but if we look at the precancerous lesions, we did develop a panel that let us look at AP CAFs relative to ICAFs and MyCAFs. And we see that all three subtypes are present in the precancerous lesions. So for the cell-cell communication, because of the culture limitations, we focused on one subset, but that's absolutely something that we're looking at. So whenever we look at any of these, right, like how do we end our biology papers, right? So we end our paper, we've done some analysis, we figured out that the fibroblasts are communicating to the epithelial cells in precancer, be it normal or tumor cells, and they're inducing a more EMT-like state. And we end with a cartoon model like this. Now, the mathematician in me is always wondering, okay, so where do we go with this cartoon model? Right? So I have this. What are the implications of this for the tumor? Right? Do we have the ability that if we've drawn this and we see a certain architecture in the tumor, can we go further and start predicting where the tumor is going to go and what are the emergent cell behaviors that would arise over time if this thought experiment of this little cartoon is what explains the biology? I'm very fortunate to have a great collaborator, Paul Macklin, who I reached out to him about this. And he's developed a software called PhysiCell that does agent-based mathematical modeling. And I'll get into what that is in a little bit. Let me see if I have that slide. No, I don't. So I'll get into it now because I don't have the slide. Apologies. So what an agent-based mathematical model is you develop a set of virtual cells that you lay out spatially on the computer. Each of those cells has an equation in it, which figures out what that cell is going to do next over time, where that equation depends both on what is the state of that cell and what are the other cells that are near it. And it has rules for how it changes forward in time. So most of these agent-based models, when you're developing them, they're very complicated to code because you have to develop the software infrastructure to lay out the virtual cells and then write the software to implement and solve for the equations within each one. So Paul has developed some of this within the PhysiCell toolkit, but it still requires manual annotation and manual selection of the cells that are there. So I was talking to Paul and saying, like, I would love to encode these types of networks that we're learning from genomics data into these agent-based models so we can get better prediction. And Paul was like, I know, I can develop a whole new grammar where I just type in plain language words what the different types of cell rules are, get the computer to parse that, and formulate different equations. And I was like, Paul, you're crazy, but okay. So 6 months later, he came back to me with the software completely developed. And so then we wanted to start sort of trying to test it and seeing what the implications were in these models. So Daniel Bergman started saying, okay, let's say that we have this hypothesis that, the cells transition from tumor to normal over time. let's say there's a stress that's induced in this EMT-like process from the fibroblasts, and we're going to model the EMT signaling as a change in motility in the cells relative to proliferative signaling that's mutually exclusive when they're away from the fibroblasts, based on this little network that we drew out. So I'm going to show you a movie in cell culture of what we anticipate would happen. So we see the cells starting to transition to be more mesenchymal-like. They move out, and then eventually what you see when it stops settling out is the cells are going to transition back to the epithelial cell type, and when they're not trapped in the fibroblasts, start proliferating and forming invasive subclones. So it gives us an ability to map that we're going to see a transition between EMT-like processes in early stages that are then going to switch to more proliferative stages as the tumor is developing, which is exactly what we observed in our precancer data. So then the question becomes, can we go one step further? And if we have spatial transcriptomics data of these tumor samples, can we see the implications of where they might go as the tumor develops, because we have the cell states mapped out in the spatial transcriptomics data sets? So we can overlay these same rules that we developed in that sort of toy that I showed you directly onto tumors. Oops, and I will let these movies actually play if they play. And we have different fibroblast density in these two tumors that are here. And so you can see the style starting to develop over time. There's a more empty space in the bottom one and more regions sort of without fibroblasts in the top. You can see them moving out and then starting to invade at the edges in the top where you have space without fibroblasts, whereas in the bottom, it's much slower to develop. And as it develops, what you're going to see is that the pockets sort of get trapped between fibroblasts. What's interesting is when you look at the invasive cells, they switch back to being more proliferative. And if you look at the subtypes of pancreatic cancer, some of the common cell subtypes are whether they're more epithelial-like or classical, or whether they're more EMT-like and basal. And everybody is associating the EMT-like state with invasion. But what's interesting is when you go back and look at the data that's actually metastatic, it returns to the classical phenotype that's more proliferative. So our hypothesis based on this math modeling is that what's happening is that the fibroblasts are inducing a transition that temporarily allow the cells to explore space and be more EMT-like, but when they actually invade, the lesions go back to being classical and being more proliferative, which is exactly what's observed in the literature. So where does this leave us, right? It's well and good to predict these outcomes, but ultimately we do want to treat patients, right? So we want to start thinking about therapeutics in this context. So the other power of these agent-based models is we can start to introduce perturbations where we can model the effects of therapeutics directly in the computer. So how are we going to get the rules of how therapeutics work? So our collaborators, Liz Jaffe and Lei Zhang, have a neoadjuvant clinical trial platform that they're running, where they're looking at combinations of vaccine and immune checkpoint inhibitors and adding in additional therapeutics to see if we can induce an immune response to tumors. In the neoadjuvant trial, treatment is applied before surgery. So then you're able to get gene expression data or profiling at the surgical time point to start looking at what's going on. Now, these trials take a long time. So this is an older technology where what Liz and Lei did is to sort out the immune cells and then do bulk RNA-seq on the sorted immune cells. So we don't have single cell data there yet. We're working on that analysis now. So what we observe if we look at the trial is that treatment, the difference between vaccine and checkpoint, is associated with your CD4 T cell response. So you have a decrease in naive CD4 T cells when you add the immune checkpoint inhibitor relative to vaccine alone. If we look at CD8 T cell clonal expansion and what's going on in the CD8 T cells, we see that survival, unfortunately, is associated with CD8 T cells, not CD4. And so we're seeing a change in the CD4s due to therapy, but not enough to be associated with a change in survival in the patients. And then if we look deeper at what's going on between the different treatment arms, we see, again, CD4 T cells, we see huge chemokine receptor binding, interesting signaling and lymphocyte chemotaxis, and then in the CD8 T cells, extracellular matrix, and then in the myeloid cells, neutrophil degranulation. So this lets us scratching our heads for a little bit about what any of this meant. And then we made a little cartoon diagram again. Okay, what if the neutrophils, as they're degranulating, are altering the extracellular matrix? And then what's happening is you see your CD4 T cells, they're secreting chemokine, which is then attracting CD8 T cells. And CD8 T cells are then having this interplay where they're seeing a change in signaling associated with chemotaxis relative to potential block from the ECM that's occurring from the neutrophils. So what if any of this is real, right? So I've given you this cartoon. This is our hypothesis. Again, this is how we ended the paper because this is what the analysis suggested. But how do we start seeing the implications of this on what it's going to do in a tumor? So Jeanette Johnson started to look at building up this model. We sort of abstracted out the neutrophils to just being ECM and start seeing how this impacts cell motility in our system. And what we observe, the immune cells are on the outside, is that the immune cells immediately start to clump together where we simulate a time point 0 giving vaccine. So Jeanette, as a graduate student, didn't show me these results for a month because she was trying to tweak the model to get the immune cells to kill, until I pointed out to her that this is pancreatic cancer. We're not going to get an immunotherapy response. And that if we look in the literature, what's been observed from vaccine is the formation of lymphoid aggregates, not an actual immune response. So we wanted to see then, as we add different therapies, can we start to see which combinations might enhance our therapeutic response? So we can go back to our trusty single cell atlas. And one of the things that's interesting with that is then we can start to look at novel therapeutic targets and say, okay, CD137 agonist is one therapy that this group is looking at. Can we start to classify cells based on their relative expression of therapeutic targets? And can we learn from the pretreatment tumors what the difference in the regulatory programs are if we would simulate sort of knocking down different genes? So our colleagues, Chris Cherry and Jennifer Alisiev, had developed this elegant ligand receptor network inference tool called Domino. And we wanted to see, could we start to find the regulatory programs that differed between cells that were high for CD137 relative to ones that were low, to see if we could estimate the impact of this therapy. And when we do this, what we observe, looking at signaling to the neutrophils, is interferon signaling that occurs in the cells, in the CD8 T cells that are high for CD137 relative to the ones that are low. And so what we hypothesized that would do is if the CD8 T cell is expressing CD137, that it could have its own chemokine signaling gradient independent of the CD4 T cells that wouldn't be there if it was low. So what if that's the case, right? Let's go back to our system and start evaluating this. So I can apply this. This is the same vaccine case I was showing you before, where we get the aggregates. If we add immune checkpoint inhibitor, the aggregates form and start to kill the tumor around the edge, but can't penetrate. If we have vaccine and urelumab, a CD137 agonist simulated, these aggregates can start to infiltrate the tumor so that when we get the triple combination, they can come in and start killing within the tumor. And Liz and Lei in the clinical studies have observed increased long-term survival and pathologic response in a subset of the tumors on their first clinical trial. It's unfortunately a phase 1, so they're expanding it out to a larger clinical trial, but this combination does seem to be one that can induce killing. So then the question always becomes, how do we validate, right? Like, how do I know if any of these cartoons are real? And I'm not going to have a satisfying answer for you guys today because this is sort of the active area of research that we're working on now. But what we're hoping is that as we get single cell data sets from these different treatment combinations, and so, for example, if we look at arm C from this trial with this triple combination, we see enhanced CD8 T cell clonal expansion in that trial arm relative to the other two, that we can start to map these simulation results back to the atlases post-treatment as it relates to therapy. So with that, I'd like to really call for thinking about moving from a precision to a predictive medicine paradigm, where we're starting to really map out these therapeutic combinations, look at them, what's going on, how they change in atlas data sets, and then have an integration with mathematical modeling so that we can start to predict how it's going to go before giving a patient a therapy. And this will probably be the rest of my career's worth of work working on this, and hopefully many other people in the audience too. Of course, this goes without saying that it really is a collaborative team science effort where I don't know how many of you saw the sort of research parasite article about bioinformaticians, but really moving away from that notion to thinking of ourselves as team members and how do we build up multidisciplinary teams to advance this type of work. So of course, I'd like to thank my funders, the members of the lab who did all the work, and amazing collaborators, and happy to take questions.
Thanks. Fascinating talk. Thank you. I have a question about the stroma business and the inflammation, and then the transition to a more proliferative state. When you model this, are those connected? Is the inflammation connected to the higher proliferative state? Can it drive it, or is this a separate thing that goes on?
We don't know yet. We definitely see, if we look and dig a little bit more deeper in the spatial data, that we can separate out the EMT signaling from the inflammatory signaling, which appeared connected in the initial analysis. I don't know what the directionality is, and that will be really interesting, I think, to look at. Jackie and Sam are going back and looking at the organoid co-culture and seeing it's very clear that the fibroblasts induce EMT. I don't know if that's as surprising of a finding as the inflammatory signaling and how it's relating.
Hi, thank you very much. Very fascinating talk. I was wondering, if we go back towards the beginning of your talk, just for my personal understanding. So basically your patterning, it's a way of creating a gene signature that is it specific for every cell type, or just takes into account the different signature that exists in different cell types? So you get like the same signature, does it changes weights across cell types, or it just cell type specific?
So it's completely unsupervised. It's a non-negative matrix factorization based approach. What we see is that some of the signatures that we see are cell type specific, and some of them, for example, like cell cycle or things like that, will span multiple cell types. So you sort of see both at once in the data.
And how robust is this approach across different, like if you take two different references, right? Two different single cell studies, do you tend to obtain the same signatures?
Yes. Yeah. We've shown previously, and we started this back in microarrays, but have moved it on. And I'm starting sort of in single cell with developmental biology. Is what we see is that the patterns that are associated with technical artifacts in the data set don't project from one data set to another, whereas the ones that are biological do. So you do get some data set specific signals, but those tend to be like batch effects or something like library depth or things like that. Whereas the ones that are associated with cell type or cellular processes seem to be more robust.
And can these signatures that stem from single cell experiment, can you apply them in bulk? Because basically what you're doing in the spatial is a pseudo bulk in a sense, like as you said, like it's not exactly single cell specific, so there is a mixture. Yeah,
you can definitely apply it in bulk and use it for sort of a deconvolution based approach within bulk to see how much you identify each within there. So that's definitely something that can be done and has. And you can go the other way. You can also learn signatures in bulk and then project them into single cell. So it really is technology agnostic. We started this project in microarrays and have just, it keeps working. So we haven't abandoned it, but.
Thanks.
For your transfer learning step, did you try like a bunch of different, I guess, pre-trained models, or did you have one in mind?
So we definitely used the matrix factorization based approach. You know, the software though that we've developed is generic, so it can use basically any gene signature method. It works for. So as long as you have a gene signature, it can project it into a new data set that's continuous. So I don't know if that answers your question, but we haven't done as much with sort of AI based, you know, like learning on these data sets with big references. We tend to explore each data set a little bit more independently to discover the nuances that are there. I don't know if that answers your question.
I think it kind of does. So you did use the transfer learning step in your model, right?
In the math model?
Yeah.
Only to label the cell types in the spatial. So we used it for the cellular labels. We didn't use it in the formation of the rules for the agent-based model. But we used it here to annotate each of the cell types for the initialization.
Was the input for that step your the results from your non-negative matrix factorization?
So the input is this spatial transcriptomics data. We have the non-negative matrix factorization applied to, so this is something I think that throws people a lot. Agent-based models are not data-driven approaches, right? And that's very important to distinguish. So this is something I think that I've seen throw people a lot, where people are very used to, in the biology community, data-driven models, right? So you're learning, if I go back to the start of my talk, right? Like my very first intro, when it stops moving, right? People are very used to, you give me a data set, I tell you what happens by analyzing that data. As a mathematician and physicist, we don't think that way typically, right? We think, what are the logical rules that can form a system of equations that abstract the system? And if I had no data at all, right? I'm in a world without any data whatsoever. Just like if I, for example, throw a ball up in the air, right? I know how gravity works and how the forces work. I can figure out how quickly that ball is going to fall without having a single data point beyond the initial velocity and our gravitational pull. A lot of times when we're building mathematical models, what we want to do is take that theory and build out what are the implications of that into a system of equations completely independent of the high throughput data. So when we develop this agent-based model, the way the data comes in is only in the formation of these rules. We learned from the data what each cell type is doing to every other one, but all we input to the model is fibroblasts induces EMT to the epithelial cell. Literally that sentence, right, as a sentence. And so that is enough to give us these types of simulations in a virtual context. What it's not is enough is to give us a simulation of what it's going to do in a tissue. So for that, we go to the spatial transcriptomics to initialize where each cell is. And our transfer learning comes in terms of annotating each cell. But then the mathematical model is still just those rules, but the initial conditions of where the cells are is determined from the spatial transcriptomics.
Thank you very much.
Yeah.
The talk. I think.
And this is, I think, where, as bioinformatics is becoming more common, I would really encourage anyone in the room who's coming from a biology background to learn about the diversity of computational sciences and learn about the diversity of computational approaches that are out there. It's not all AI, it's not all data-driven, and it's really important to understand the role that different computational models make in the inferences that you get from your data.
Hi, thank you for the talk. Did you ever simulate out or, I don't know, observe any differences with the macrophages?
We haven't done that yet. That's something that I have a graduate student who's starting to develop the macrophage co-culture, so we can do that. So that is coming. We have a little bit where we have a model that's not grounded in data, but with Lisa Cousins, where we've been looking at macrophages and how they can be compensatory with T cells. So we have modeled that to some extent, but we need to go back and get the experimental data to validate that.
I have a question. So I think this agent-based forecasting is really interesting. And kind of like you said, it's not very commonly seen in the space. I guess I'm curious if you think this is a good long-term solution to the problem that you're facing. And do you think, although it's not data-driven, do you think you could update the equations as more single-cell data is acquired over time and make them more accurate? And then, of course, you said the validation is necessary, and I'm really interested to see kind of how it does perform against some ground truth.
Yeah, and we were talking about this on the walk over too. So this goes back to my weather days too, is one of the things there. In these complex systems, you can't predict them forever, right? There's a limit to how long you can predict them. So the way our weather forecasts work and deal with this is that they keep sort of stopping the model and integrating when new data come in to update. So I think we're going to have to get to a mode like that in this if we're ever going to apply these sort of approaches in any sort of clinical or real world setting. The issue is, how do you actually get time course data in patients is, unless you're working in blood tumor, is almost impossible to do in a non-invasive way. So thinking about when, I think that there's some very fundamental questions that we need to ask as a field in terms of, when do patients need to come back for new data sets? What type of data is needed at the next time points, and how do we use that we just fundamentally don't know.
Maybe mouse models potentially could.
Mouse models, organoids, I think those will start getting us there, but there's a lot of additional complexity in humans.
Oh, I guess kind of Yuan Chen on
I think
Zoom
there's a
asks,
question in the back as well.
Oh, well, I guess someone asked, Dwayne here, I guess Yuan kind of asked the same question I just said about mouse, but Dwayne asks, do you correlate differences in gene expression with perceived differences in local resources like hypoxia, nutrients, etc.? Would this be useful, or does it all come out in the wash?
Yeah, no, I think it's really useful. Hypoxia is a tricky one because you end up with necrotic cells and things like that, and how you handle that. But absolutely, being able to correlate it with nutrients, depending on what you look at, is going to be really important.
Yeah, I want to follow up on the organoids. I have a collaborator that I work with, and we do prostate cancer bone metastasis cells passaged in mice, and so they're human cells,
Cool.
passaged in mice, and also organoids. So we have them in time series. And it'd be really interesting to use this kind of approach to predict what the next organoid is going to do. Have you thought about how to use organoids?
Yeah, that's a good, I haven't thought about it in that context. We've been thinking about it in terms of the inflammatory, like the EMT-like signaling, like is it related to a cell motility? And can we model that more shorter in time in therapeutics? But I haven't thought about it in the context that you're bringing. It would be really interesting to see if you could predict the states in them. That would be cool.
You could predict and then measure. And I really like the weather analogy, this idea that with a modeling system, and maybe a question to clarify, when you have this modeling system, is it dependent on the start state? So could you use a patient's current data as the start state and predict the weather in that tumor?
Exactly. And that's exactly what this case is, right? Like that's exactly what I'm showing you here. So that's very much where I hope we go, is that we'll initialize to different patients. And I think the question is, how sensitive is it to the patient's initial conditions? How much does it change over time? And I really do think of it as more than an analogy. I know people think I make this huge research shift, but to me, this is like all the same. Mathematically, it's the same thing that I'm trying to do, just a different system.
Have you considered, in terms of like validation data, using a single sample and trying to understand like a trajectory within the sample, and then trying to predict the trajectory by like taking like the community at some point in the trajectory and using that as your initialization?
Yeah, I mean, that's an interesting idea. I think we are wide open to how to validate in this context. Like I said, we've been thinking about it mostly like, can we the simplest thing I can think of is, can you simulate a perturbation? And then do you get the accurate simulation of a perturbation? That's a lot of how we've been thinking about it. But developing the framework as to how we estimate and what the right quantification for validation is, and like, do you do it? So for example, in the precancer, we've been thinking if we get low grade and high grade that are derived from the same patient, could we use that to monitor? If we know that they're genetically related, that's another way. But yeah, certainly some of these organoids are probably even simpler, and we should probably chat and actually think about that.
I just wanted to say that at the peak of attendance, you had 55 people in Zoom, and I think even more here. So thank you for
Thank you.
attracting a great audience.
Thanks for having me. It's my pleasure.